In this presentation, I would talk about using cyclosal prodrug for improving absorption of stavudine drug, which used to treat HIV infection, also effective anabolism, and a poor catabolism cannot always be predicted in phosphorylation for antiviral drug. As we all know that the mode of action of most of the nucleoside analogs occurs through the inhibition of DNA polymerases. Intracellular conversion of the nucleoside analogs into their 5-triphosphates is a prerequisite. D4-TMP, catalyzed by thymidine kinase, TK, is a critical rate-limiting step in human cells. Nucleotides are charged molecules under physiological conditions and do not efficiently penetrate cell membranes. This difficulty can be surmounted by attaching suitably degradable lipophilic groups to the phosphate moiety that leads to neutral, membrane-permeable, masked nucleotides. The synthesis of the cyclosal pronucleotides has mostly been achieved by using chlorophosphate or phosphoramidites. Generally, diol 6 were reacted with PCl3 to give chlorophosphate 8 that were reacted with the nucleoside analog in the presence of disopropylethylamine to yield the cyclic phosphate triesters that were oxidized in a one-pot reaction using T-butyl hydroperoxide. They used salicyl alcohol as a cyclic bifunctional masking unit in the pronucleotide approach. In the hydrolysis process, first the phenolate is displaced preferentially at the phosphate group, leading to 2-hydroxybenzyl phosphate, diester 5. The remaining masking group is activated and a spontaneous breakdown of diester 5 to the nucleotide and salicyl alcohol 6. It has been observed that a cleavage of the benzyl ester bond to give diester 7. I must mention here that salicyl alcohols 6 were tested for their biological potency, but showed neither antiviral activity nor cytotoxicity, which very important consideration on prodrug formation. Extensive studies have been performed in order to prove the delivery mechanism of D4-TMP from cyclosal triesters. Half-lives depend on the substitution pattern of the aromatic ring. Acceptor substituents cause a decrease in hydrolytic stability, while donor substituents, methyl and particular T-butyl group ES, led to an increased stability. All data obtained from hydrolysis and NMR studies are in perfect agreement with the designed degradation reaction mechanism. Also, they have shown that the discussed hydrolysis pathway is independent on the nucleoside analog attached to the phosphate moiety. The results showed comparable or even higher antiviral potency in a wild-type T lymphocytic cell line, CEM/O, compared to D4-T1 period, and which is good about cyclosal approach, needs only one bifunctional masking unit per equivalent nucleotide molecule. Thank you.